Yes, ladies and gentlemen, we would like to welcome you to lesson 2. Our topic is nutrition, and today we are discussing about food. And this is not new to us. In this particular lesson, we are going to look at the definition for food. We shall also look at the different types of food that we consume, and then we shall also look at the chemical tests for these particular food substances that we would have looked at. So let's start with the definition for the food. So members, food is any substance which can be digested and absorbed by the body to maintain the body's life processes. We know very well that our bodies run very many different processes every time. So those processes require food to enable them run normally. The processes that occur in the body are called metabolic processes. So, can we all define food now? Food is any substance which can be digested and absorbed by the body to maintain the body's life processes. So, if you are going to take break today, or lunch, or supper, you know that what you are taking is very vital for the processes that take place in our bodies. So I want to look at the requirement for food. Why do we require food in our bodies? To begin with, growth. Food is required for growth, so as to build new cells. We know that in our bodies there is a process of cell division, which leads to increase in numbers of cells and eventually leading to growth. So that process there requires raw materials from different food sources. Secondly, we also require food for the process of respiration in order to produce energy that we require. Energy which is used for thinking, for muscle contraction, even for cell division, and for many other body activities. Food is also required for repair of worn out cells and tissues in the body. So all the time there are very many tissues and cells that get worn out. For example, we have the red blood cells. Their lifespan is 120 days. So every 4 months, we have to replace our red blood cells. So the production of red blood cells requires some materials, from the food sources that we eat. The other one is protection of the body against disease. We know that there are some foods that we eat in order to boost the immunity of the body. We eat vegetables, we eat fruits, and so on. And in those sources, we get the vitamins and the other foods that are required for protecting the body against infections. So at least we now know the reasons why we require food in our bodies. What would happen if you do not eat food? What is the consequence to the body? If you don't eat food, it means that the processes we have mentioned there, process of growth, respiration, repair, and protection of the body against disease, will not take place. And if those processes don't take place in our bodies, it simply means you will be as good as dead. Let's look at the classes of food. Food is classified into 3 major classes, and these ones include the energy-giving food. There are those foods that are called the energy-giving foods. We know them. Those ones are majorly carbohydrates, and they are lipids as well. Fats and oils are the ones we call the lipids. And then we have another type of food, another class called the bodybuilding food. These foods are sometimes called the growth food, and they are majorly the proteins. These ones are very essential for the building of new cells in the body, so as to increase in size. The other type or class of food is the protective foods, and these protect the body against infections and diseases. and this includes the vitamins and the minerals. So members, there are three major classes of food: the energy-giving foods, the bodybuilding food, and the protective food. Those are the three major classes of food. So let's look at these foods much more in detail. We have the energy-giving foods, the ones that are called collectively as carbohydrates, fats, and the oils. You know very well where we can get them. The sources of carbohydrates include rice, maize, bread, chapatis, potatoes, bananas, and so on. Those are the sources of the energy-giving food. Those are some of the sources, and there are very many other sources. I want you to name other 5 sources of the energy-giving food. What else do you eat to get energy apart from those that we have named? I want you to give me other 5 foods that you eat. The other one is the bodybuilding food. So let's look at some of the sources of these foods. Where do we get them? These bodybuilding foods. If you want to get the bodybuilding food or the proteins, what do you need to eat? So we have some of the samples there. We can eat eggs. Eggs are very nutritious. They have proteins. We can eat chicken. We can eat beef, fish, soya beans, or even beans. We have very many others, milk and so on. So I want you to continue naming them. At least give 20 foods that you can eat to get proteins. Give me other 20 that you know, apart from the ones we have named there. And then we have the protective food. These ones basically protect the body against infections and diseases. And these ones are majorly the vitamins and the minerals. And we get them from fruits and vegetables majorly. So you can see some of them there where you can get the protective food. So if you eat tomatoes, you eat oranges, you eat other vegetables, then you are very sure that you are getting protected against disease and infections. So they are very good food, and they are sometimes called the immune boosting foods. So I want us to look at the types of food, nutrients, compounds in our bodies in detail now. So members, there are 6 different nutrient compounds in our bodies, and we are going to name them. 6 different nutrient compounds in our bodies. And already we have mentioned some of them in the groups that we have given. Remember, the other ones are categories of food. There are 3 major categories where we had bodybuilding food, we had protective food, and energy-giving food. These ones are nutrient compounds. Yeah? So we want to look at the different nutrient compounds that we desire in our bodies. The first one is carbohydrates. The second one is proteins. The third one is vitamins. The fourth one is mineral salts. The other one is roughages and water. And then we have lipids, or fats and oils. So there are 6 different nutrient compounds, and all of them are very vital to support the metabolic activity in our bodies. So I want us to start by one. The first one we said was carbohydrates. And remember, in the categories of food, we had said that carbohydrates are also called energy-giving foods. Well, let's look at them now in much more detail. So these are made up of carbon, hydrogen, and oxygen. So when you look at their chemical composition, you realize that they contain majorly 3 elements. The element called carbon, the element called hydrogen, and then oxygen. And they are either sugars or starches. These carbohydrates are also grouped into 3 categories. There are those ones that are called monosaccharides, that we shall look at later on. Then there are those that are called disaccharides, and then there are those that are called polysaccharides. polysaccharides. So if you are asked, give 3 categories of carbohydrates, you should be able to ably give the 3: monosaccharides, disaccharides, and polysaccharides. And this grouping is dependent on the number of sugar molecules that make up these carbohydrates. If there is 1 sugar molecule, we call them monosaccharide. If there are 2 sugar molecules, they are called disaccharides. If there are many, they are called polysaccharides. So let's start with the first one, the first category of carbohydrate, monosaccharides. And the word mono means 1, and saccharide means sugar. So it simply means these monosaccharides consist of 1 molecule of sugar. And they are also called simple sugars. Of course, they just contain 1 sugar molecule. So their structures are very simple. So let's look at their properties. Being simple sugars, what would you expect from the monosaccharides? One is that they are sweet. They have a sweet taste. Yeah, they have a sweet taste. So some of the things that you eat that taste sweet may be containing monosaccharides. The other one is that they dissolve in water, meaning they are highly soluble in water. They also form crystals. I hope you know what crystals are. When you touch them, you feel small solid particles. Those ones are called crystals. So monosaccharides form crystals. The other one is that they can pass through selectively permeable membrane. Yeah, they can pass through selectively permeable membrane because of their small structure. Yes. And then the other characteristic is that they can change the color of Benedict solution from blue to orange. Yes, when you boil it with Benedict solution, meaning they are reducing sugars. That means they are capable of reducing the copper 2 ion in Benedict solution to copper 1. Copper 2 is blue and copper 1 is brown or orange. So they are reducing sugars. So the properties of monosaccharides include they are sweet, they dissolve in water, they form crystals, and they pass through selectively permeable membranes, and then they are reducing what? Sugars. That's great. Then let's look at the monosaccharides, and now the examples of monosaccharides. The first one is glucose, which you seem to be familiar with. And then we have fructose. You can pronounce it as fructose or fructose, no problem. And then we have galactose. So there are 3 major or 3 common monosaccharides: glucose, fructose, and galactose. However, there are others as well, but these ones are the major ones that we usually interact with: glucose, fructose, and galactose. So those are the monosaccharides. Let's look at now the other second category of the carbohydrates, and this one is the disaccharides. Remember, the other one was monosaccharides. This one is the disaccharides. So disaccharides comes 2 words: di meaning 2 and saccharide meaning sugar. That means these carbohydrate molecules are made up of 2 simple sugars joined together. So these simple sugars, or these monosaccharides, join together through a condensation reaction to form what we call disaccharides. So when the 2 monosaccharides combine, it results into loss of 1 molecule of water, and this reaction is called condensation reaction. So here it's not so clear, but you can see there it is C6H12O6 plus C6H12O6. So that is glucose. 2 glucose molecules combine to form maltose. So glucose and glucose, those are monosaccharides, and then those 2 combine to form maltose, which is now a disaccharide plus water. What about if we combine glucose and galactose? Those 2 are also monosaccharides. Glucose and galactose, we form lactose. Yes, we form lactose, which is a disaccharide. Then if we combine glucose and fructose, what would we form? We form sucrose. And sucrose is the common sugar that we take at home, the table sugar, and it is a disaccharide. So that's great. So when we have three major or three common disaccharides. We have maltose, which is made from glucose and glucose, two glucose molecules. Then we have lactose, which is made from glucose and galactose. And then we have sucrose, which results from a combination of glucose and fructose. So let's look at the properties now of the disaccharides. One, they are sweeter than monosaccharides. Remember we said monosaccharides have a sweet taste, but disaccharides are sweeter. Yes, I want you to do this experiment. You buy a glucose in the shop, buy that glucose in the packet, and also get a spoon of sugar at home, which is sucrose, which is a disaccharide, and test them one after the other. You can first test glucose, put it in your tongue and taste, and also get a spoon of sugar later and test it as well, and compare the different types of sweetness. So you may realize that the disaccharide sucrose, which is table sugar, is sweeter than glucose. You can try that at home just to confirm that point. The second one is that they are crystallized, or they are crystallizable. They can form crystals. And when you touch sugar with your fingers, you feel those particles, the salty particles. Those are the ones we call crystals. They are also soluble in water, and that's why we are able to form, to make tea. Yeah, you add sugar to hot water, and you are able to get tea, which is sweet. And then for them, they do not change the Benedict solution when heated with it, apart from one of them, which is maltose. So that means they are non-reducing sugars. It's only maltose which is a reducing sugar. The rest of the disaccharides are non-reducing sugars. They are unable to reduce copper 2 in Benedict solution to copper 1. Yeah, that's great. Then we have, they can also be broken down to simple sugars by dilute mineral acids and enzymes. So these disaccharides can be broken down into monosaccharides. One disaccharide can be broken down to produce two monosaccharides. For example, maltose can be broken down to produce two glucose molecules. Lactose can be broken down to produce galactose and glucose. Sucrose can be broken down to produce fructose and glucose. And this can take place by action of enzymes naturally. Even in our bodies, this takes place naturally. Enzymes break them down. Yes. And then, for example, if it is maltose, there is an enzyme called maltase, which breaks maltose down to two glucose molecules. If it is lactose, there is an enzyme called lactase, which breaks down lactose to galactose and glucose. If it is sucrose, there is an enzyme called sucrase, yes, that breaks down sucrose to fructose and glucose. And we shall look at the enzymes later on. And the process by which a bigger molecule is broken down to smaller molecules, in this case, is called hydrolysis. That process is called hydrolysis. Okay, we have just looked at disaccharides. So I want to welcome you to the third category of carbohydrates called polysaccharides. The word poly means many, and saccharide means sugar. So simply, this is a very complex carbohydrate made up of many units of simple sugars. So there are very many, they combine to form polysaccharides. They combined, very many monosaccharides combined. They undergo condensation reactions to produce polysaccharides. And let's go to their properties. They are not sweet. Yeah, polysaccharides are not sweet. Remember we said disaccharides and monosaccharides are sweet, but polysaccharides are not sweet. They do not dissolve in water. They cannot be crystallized, meaning they don't form crystals. For them, if you grind them, they form powders. They do not change the color of Benedict solution, meaning they are non-reducing sugars. Examples include starch, glycogen, cellulose, chitin, and so on. Those are some of the examples of polysaccharides. Yeah, I want to ask you to give me some of the sources of polysaccharides. Where do we get polysaccharides? For example, starch. Which food do you eat in order to get starch? Please write for me those answers and send them on my inbox. So let's look at the functions of carbohydrates in general. The polysaccharides, the monosaccharides, and the disaccharides. Let's look at their functions. One, they provide energy. They provide what? Energy in the body when oxidized during respiration. So when you oxidize them during respiration, you are able to get energy, meaning they are energy-giving food. They are cheap sources of energy for living things, of course, that's what we have mentioned. They also act as food reserves, which are stored within organisms. E.g., many plants store food as starch, and animals store them as glycogen. So in many living systems, usually these carbohydrates are stored as food reserves, such that in case of any deficiency, these reserves can be broken down and utilized accordingly. They are important components of body structures. For example, cellulose is a component of cell walls in plants. Chitin forms exoskeleton of arthropods, and heparin is an anticoagulant in mammalian blood, which prevents blood from clotting, which keeps the blood liquid when it is flowing in blood vessels. That heparin there. So they form different structures in the bodies, and they are very vital structures. They are important for commercial values. Yes, for example, they provide raw materials for the manufacture of various products. For example, cellulose provides raw material for manufacture of paper and textiles. Yes, so those are some of the functions of carbohydrates that we need to know. So carbohydrates, therefore, are very important. I think their most important function is they are energy-giving food. If you wanted to know carbohydrates, please you would easily know them as a food that gives you energy for doing work. So what happens if you don't have carbohydrates in your body for long? If you lack carbohydrates in the body, you will suffer from a condition called marasmus. So deficiency of carbohydrates results into a deficiency disease or syndrome called marasmus. So how do you know that you may be having marasmus or somebody might be having marasmus? The first one is high appetite. So those people have a quite higher appetite compared to others, compared to normal. And then the dehydration of the body, meaning the body will not have water. There will be little water in the body, and there will be dry eyes, dry lips, and so on. Yes, that one, most likely somebody might be having marasmus, because these sugars are responsible for water absorption in the body. So if they are not there, then you are likely to suffer from dehydration, which is a condition caused by lack of enough water in the body. Growth retardation. So those people have stunted growth. Yeah. Then wastage of muscles. The person grows smaller and thinner. There is loss of weight, therefore. There is also misery and sunken appearance. So there is unpleasing appearance from the person who is suffering from marasmus, as you may see there. We hide the face for protective purposes, but you can see the body. Well, food tests on carbohydrates. Now that we have seen the carbohydrates we looked at the monosaccharides, we looked at disaccharides, we also looked at polysaccharides. And we are going to do chemical tests to prove whether the foods have these different carbohydrates. And carbohydrates are categorized into three major. In our level, we test for three major carbohydrates. We test for reducing sugars, we test for non-reducing sugars, and we also test for starch. Those are not the only carbohydrates categories. There are many others, but those are the ones that we can easily test at our level. So let's start with the first one, test for reducing sugars. So this is a chemical test in the laboratory that we can conduct to confirm whether the food source has reducing sugars. So this is what we do. What we use is a reagent called Benedict solution, and it is usually blue. If the Benedict solution is not there, we can use the Fehling solution, which serves the same purpose, and it's also blue. For this test to be successful, you may need some heat, so boiling is required in the course of the test, as we shall see. So at least for now, from this slide, what I want you to realize is that the chemical that we use for testing for reducing sugars is called Benedict's solution or Benedict's reagent, and its color is blue. If you don't have Benedict's solution, you can use Fehling's solution, and its color is also blue, and boiling is required. So that's what I want you to know from this particular slide. Benedict is the name of a person, so the first letter starts with a capital B. Then even Fehling's, the same story. Whenever you're writing them, you start with capital letter or uppercase. So this is what the procedure, the observations and conclusions would look like in case you went to the laboratory. The procedure is as follows. These are the steps you follow. To 1 cubic centimeter of a food solution that you don't know, maybe X, maybe A, as you may be given, you add 1 centimeter of Benedict solution and boil. So it's very simple now. You just get 1 cubic centimeter of a food solution in a beaker or whatever, how they have given you, put it in a test tube, and then you add Benedict solution, also the same quantity, 1 centimeter cubed, and then you boil. Usually you use a test tube holder, and then you can boil either with the water, from a water bath or from the test tube. Of course, care must be taken as you're boiling, as a teacher will guide you. Then what would you expect when you do that? So depending on what they have given you, it can be a colorless solution, it can be a turbid solution, so whatever, it will turn blue. Of course, you said Benedict solution is blue, so when you add Benedict solution to that food solution, the color of Benedict will be seen, so it will turn blue solution. And then it will turn to, when you boil it, it will begin changing colors. It will turn to green solution. Green is a solution. It will turn to yellow precipitate. You will start seeing a precipitate. It will turn to orange precipitate, and finally to brown precipitate. So what does that mean? Each color change there means something. If the solution turned to blue, of course, then on boiling it turned to green. It means little reducing sugars are present. If you boil and it remained green, little reducing sugars present. If it turned to blue and then to green solution and then to yellow precipitate, it means moderate reducing sugars are present. Moderate, not much, not little. Moderate reducing sugars are present in that solution. What about if it turned to blue on adding of Benedict solution? Then on boiling it turns to green. As you continue boiling it turns to yellow. As you continue it turns to orange. What does that mean? And remains orange. It means much reducing sugars are present. What about if it continues to brown precipitate? It means too much reducing sugars are present. So it's as simple as that. Yes, as you boil, you keep on observing the color changes. These color changes take place very fast, so you must concentrate as you are boiling. Do not play, don't look aside, just concentrate on your test tube. You will be able to see all the color changes. And you slant your test tube a bit, you bend it. That's when you can be able to see all the color changes clearly. So what about if the colorless solution or the turbid solution turned to blue solution? And then when you boil it remains blue, no change. It means, what does it mean? Reducing sugars are absent. So that's how we test for reducing sugars. I want to task you to rewrite the procedure on your own. Write the observation and also write the possible conclusions on your own in your summary book. Please do that and then send me your responses in my inbox. So let's look at more facts about this test. Now, if failing solution is used, the change is from blue solution to orange precipitate if the reducing sugars are present. It remains blue if they are absent. So it's the same as the test for Benedict's. The failing test is the same as the test for Benedict's test. So it is the same. So what are the reducing sugars we are talking about? What are those ones? We're talking about glucose, which is usually present in the grapes. We're talking about fructose, which is majorly in fruits. You have tasted many fruits and they are sweet. Bananas and so on. They contain some bit of fructose. They were talking about galactose and maltose. Those are the reducing sugars that are commonly found in our environment, and they are the ones that we can mix and form that solution, and then you will be given to test. So what are the conclusions now? The conclusions based on the color changes according to the following observations. If the solution remains blue after boiling, it means no reducing sugars are present. If the solution turns from blue to green and remains there, it means little reducing sugars are present. If it turned from blue to green to yellow precipitate, it means moderate reducing sugars are present. If it changes from blue solution to green solution to yellow precipitate to orange precipitate, it means much reducing sugars are present. From blue solution to green solution to yellow precipitate to orange precipitate to brown precipitate, it means now too much reducing sugars are present in that solution that you are testing. So that is how our conclusions will be. You can only get one of those conclusions. You cannot get more than one. You can either get when it is only no reducing sugars, when they are little, when they are moderate, because the same solution cannot have double standard of solutions. It's not possible. So you'll watch out and see which one you have achieved, and then you record with good handwriting, and then you present your work for marking. Great. The second category of sugars we want to test are called non-reducing sugars. They are called what? They are called non-reducing sugars. Meaning these ones are not able to reduce copper 2 iron to copper 1 in the Benedict solution. So let's see what happens to them. So what do you do then? Let's see what happens. So the procedure, if you want to test for non-reducing sugars, this non-reducing sugar test always comes after the reducing sugar test has been conducted. In case you have conducted the reducing sugar test and sometimes you find it negative, no reducing sugars present, you may be tempted to confirm whether non-reducing sugars are present by conducting this other test. So the procedures, but also of course you follow what the examiner has told you or what your teacher has told you. So this is what you do. In this test, 1 centimeter of a food solution, you add 1 centimeter cubed of dilute hydrochloric acid. Or sometimes you can be told to add drops, maybe 5 drops of dilute hydrochloric acid, and then you boil. And then after boiling, you cool under water. You put it in cold water, the test tube. And then you add 1 centimeter of sodium hydroxide solution after cooling, followed by 1 centimeter cubed of Benedict solution. So let me repeat that procedure. To 1 centimeter cubed of a food solution, maybe X, Y, or P, whatever, you add 1 cubic centimeter of dilute hydrochloric acid and boil. After boiling, you cool. You run it through cold water, but not immediately after boiling. You leave like a few seconds after boiling, and then you run it through cold water to cool it. And then you add 1 centimeter cubed of sodium hydroxide solution, followed by 1 centimeter cubed of Benedict solution, and then you boil. Yes, and then you boil. Usually for 30 seconds or for 1 minute there. You'll be able to see all the observations you are supposed to see. So what would you see? A colorless solution or a turbid solution, you write 1 depending on what you have seen. Turns to a blue solution. Of course, when you add benedict solution, it turns to a blue solution, then to a green solution, to yellow precipitate, to brown precipitate on boiling, depending on the quantity of the sugars present. So it can be little, moderate, or much, or too much non-reducing sugars present. I mean, it is not non-reducing, it will be reducing sugars present. It will be little reducing sugars present, moderate, or much reducing sugars present. You remove the word none there. We shall explain later on. Now, these reducing sugars are present because the non-reducing sugars have been hydrolyzed. Yes, have been broken down to produce reducing sugars. The reason why we add hydrochloric acid is to break down the non-reducing sugars to reducing sugars. And then if the solution remains turbid, I mean the solution of a colorless solution or a turbid solution remains blue, and when you boil it remains blue, it means the reducing sugars are not there. It means reducing sugars are absent. Or you can say the non-reducing sugars have not been converted to reducing sugars, or non-reducing sugars not hydrolyzed to reducing sugars. Yes, so reducing sugars absent because non-reducing sugars not hydrolyzed to reducing sugars. So from that conclusion, I mean from this table, we can explain further and say that the non-reducing sugars actually do not have a chemical test. There is no chemical test for non-reducing sugars directly. But what we do is we hydrolyze them. We break them down to produce reducing sugars. And then we test the reducing sugars that have been produced using the Benedict's test. Yes, we carry out the Benedict's test on the reducing what? Sugars. So let's explain other facts of the test here. When boiled with dilute hydrochloric acid, when we boil this solution with dilute hydrochloric acid, what are we doing? We are actually breaking down the non-reducing sugars into reducing sugars. We are trying to break down, for example, sucrose into glucose and fructose. And then why do we add sodium hydroxide solution or sodium hydrogen carbonate powder? Is to neutralize the hydrochloric acid that we have used. So that's why the quantity of sodium hydroxide and hydrochloric acid usually is relatively the same, or sodium hydroxide is slightly more. Yes, slightly more to neutralize hydrochloric acid fully in the solution, because the Benedict solution works best in a neutral medium. So the examples of non-reducing sugars include sucrose, lactose. Those are the major examples of the non-reducing sugars, sucrose and lactose. There are others, but those are the major ones that we usually test for. So that one is the test for non-reducing sugars there. So let's look at the last test here, for test for starch. This should be the simplest test for you in the laboratory. So the procedures are: to 1 centimeter cubed of food solution, add 3 drops of iodine solution. So just get a food solution which is there, a suspected starch solution. Add 3 drops of iodine. 1, 2, 3. What would happen? That solution can be colorless, turbid, or cloudy. So you can say a turbid solution turned to a black solution, or it could have turned to a blue-black solution, or to a blue solution, or brown solution with black specks. Now I have given you all this because there are possibilities in this test. So we shall explain what each one of them means. Now if the colorless solution or a turbid solution turned to a black solution, it means much starch is present. After adding iodine turns to black, it means much starch is present. If it turns to blue-black, it means moderate starch is present. If it turns to a brown solution with black specks, the solution is generally brown, but you see some black dots within, suspended around the solution. It means little starch is present. It is only one of those possibilities that you will see. You cannot see all of them. So you only report what you have seen. And then what happens? What about if the color solution or a turbid solution turned to yellow or brown? You can only see one color there, either yellow or brown. You report only one that you have seen. That one would mean that starch is absent. But usually you would see it yellowish, or it turns to yellow, because the starch, which is brown, will be diluted and will look yellow. But you report what you have seen. If you have seen it brown, you write it there. It means starch is absent. So members, we have carried out tests, 3 food tests. We have tested for reducing sugars using a Benedict solution, and the condition is boiling. We have carried out a test for non-reducing sugars, where we first had to hydrolyze the non-reducing sugars using hydrochloric acid, and then we neutralize it, and then we carry out the Benedict's test for reducing sugars. And then we have also carried out a test for starch. I want you to rewrite these tests in your summary book. Write the test for reducing sugars, for non-reducing sugars, and for starch in your book. Write the test procedure, the possible observation, and the possible conclusions or deductions. And then you send me that summary I would want to see. If you did that, you would be a very good student. So that when you go back to school, you will be able to conduct these experiments very successfully. I want to thank you very much for being very attentive, and I want to continue encouraging you to stay at home until the school is safer for us to return. Thank you. Stay